Let's get to know our world. The Human Genome Project is one of the biggest steps that humanity has taken in the path of self-knowledge. With this project, it is aimed to map the base pairs in human DNA and understand human genes within the framework of an international and joint research program. It was completed in April 2003, allowing us to understand the genetic nature of humanity for the first time. On our planet, where more than 7 billion people live, each individual consists of more than 60 trillion cells. About 30 trillion of these cells are human cells. About 30 trillion are microorganisms living in the human body. These harmless microorganisms, which can be viruses, bacteria or fungi, are called microbiome, microbiota, and are most commonly found in the intestine and skin. So how do our cells know what to do? How they work together to help the heart pump blood to our body, play a role in our thinking, or the growth of our bones, and they know their role. Of course, the answer to this lies in what we describe as deoxyribonucleic acid. DNA resides in every cell of ours that takes part and shows them what to do. Researchers refer to the instructions for all this DNA as genome in general. What is DNA? How does DNA damage affect cancerousness? What is the task of our genome? There are 2 copies of DNA in the nucleus of each cell, one coming from the mother and the other from the father. Our DNA creates chromosomes by being folded, packaged in a special way. Inside our cells are chromosomes with 46 bundled DNA strands, 23 from the mother and 23 from the father. With these chromosomes, we develop from an embryo to an adult of about 30 trillion cells. But DNA doesn't just mean growth, it also controls what cells have to do. They also control what cells have to do in the face of ingested foods, microorganisms, or toxic substances. To understand DNA, let's look at its structure. DNA is a long, lined polymer of molecules called nucleotides that hold together. 4 different nucleotides make up DNA: adenine, guanine, cytosine, and thymine. Adenine binds with thymine and cytosine with guanine, so the 2 nucleotides linked in this way are called one base pair. Depending on the distribution of these base groups, DNA can produce many different types of organisms. We can say that these bases are letters, the distribution of the base groups are words, and the sum of these words make up the book of humanity, that is, the genome. Scientists are trying to reveal the algorithms of our diseases or differences according to the letter errors, sentence deficits, or the arrangement of the elements in these words. A human has approximately 3 billion nucleotide and 1.5 billion base pairs, and for every sperm and egg produced, these 3 billion DNA letters must be copied and packaged so that they can be passed on to future generations. Are we all different genomes? Of course, people are in very different shapes and bodies because of their appearance. We can consider different eye, skin, hair colors, or gender differences. However, when we look at the DNA level, humans are 99% similar to each other. That 1% rate makes us different from each other. This 1% difference affects us from our appearance to our risk of developing diseases and even our drug resistance. So can we say that everything is determined by our genomes? No, because DNA is such an incredible piece of the puzzle that makes up us. When we think in terms of our health, we add our lifestyle and environmental factors to this puzzle. If we know the reaction of our genome to these environmental factors, we can shape our lives in that direction. What are genetic mutations and how do they cause diseases? The tiny differences in our genomes make us unique, that is, they separate us from other individuals. Most of these differences in our genomes have little or no effect on our health. But of course, it is not always like this. If some information is erased or misspelled from our DNA, it can cause the proteins that are the product of the information to be produced damaged, overproduced, or not produced at all. These changes are called mutations. Genetic mutations can cause serious health problems. because they affect the most important building blocks of our body, namely proteins. For example, we can count proteins, the way our cells maintain their shape, the production of enzymes necessary for digestion, and the transport of oxygen in our blood. The passing of germline mutations, a special type of genetic mutation, to the next generations explains how some diseases are passed down in families. If you have a particular disease in your family, your doctor may tell you that there is a family history problem here. So what does genome research mean to you? When we looked at most of the previously produced drugs, the main idea was that drugs were equally effective in all people. However, genome research made it possible to think differently in this area. Compared to other people, according to your genetic condition, the drugs have been shown to cause more or less of them, faster or slower. Thanks to genome research, doctors will soon be able to recommend the right drug and the right dosage to be more effective than our genes. These researches will also enable the production of more sensitive, personal medicines. Instead of imaging diseases based on their response to given chemicals, researchers are now working on drugs that target specific metabolic pathways of diseases. In this way, it is hoped that more effective results with fewer side effects on diseases will be achieved. Some efforts for gene-based drugs in leukemia and some cancer treatments have already started to use. Isn't it time for a more sensitive medicine? Cancer and precision medicine. But these are not the only benefits of genome research. In the coming years, some genetic tests are expected to predict diseases. Thus, the judgment that one treatment is valid for all will come to an end and healthcare professionals will focus on personalized diagnosis, treatment and prevention. If there is a risk of colon cancer for you, you will probably be able to get advice to reduce your meat consumption after a colonoscopy. Or, if a risk for heart disease is predicted, you may need to exercise more frequently and start medications to lower your cholesterol, according to your genome information. What are the social and ethical problems posed by genome research? While research on the Human Genome Project continues, some important social and ethical problems arise. If we look at these problems from 3 different points, 1. First of all, what genetic information should be studied and who should have control of this information? Advanced diagnostic techniques can provide very important information about the individual, but this can cause serious ethical problems for individuals, employers or different departments such as insurance companies, health consulting professionals. 2. Second, what genetic procedures should or should not be performed? Should techniques such as gene therapy or enzyme therapy with modifiable human genotypes and phenotypes be prohibited or restricted? 3. Third and last, how will this information change lives? To what extent are people prepared for the findings of genetic research on human attitudes and behavior, and will this information that humanity encounter in the process of self-knowledge cause a struggle? If it does, will social institutions be able to intervene adequately in conflicts in this area? National Human Genome Research Institute in 1990, he started a program to investigate the ethical, legal and social effects of genome research and to come up with solutions. This research program, known as ELSI, is currently underway.